Again, good afternoon and welcome to our class in ATC 111 Crop and Animal Improvement. So, last time was holiday, so we haven't talked about plant genomics, but I hope you have read in advance the overview, the link that I've sent to you. So, this one, yeah, I remember. Humana man din nato din, no? LC. So I remember I have mentioned to you this one last time and maybe we're just cut off. So this, I hope you just have read through this one, although you don't really understand very well. So I think nag-stop ta diri somewhere sa EST or Express Sequence Pad. So this is uh, another what do you call that, sequencing facilities, no, or an advancement on genetic modification. So this, we have here a figure on how EST or enhancing is done. So and these are plants or species with their EST's number. So you will have an idea how many EST or express sequence tags they have done. So we have gone through with this one, and we also have the so-called reverse genetic or traditional genetic analysis, which aims to identify DNA sequences associated with a given phenotype. So by reverse genetics, it determines the function ng gene for which the sequence is known by generating and analyzing the phenotype of the corresponding knockout mutant. So meron tayong knockout mutant na parang this will generate and analyze the phenotype, no, the sequence itself. So again, we have mutagenized population ng Arabidopsis, which is used, no, and there are two main possibilities in the gene tagging and insertion side. So meron tayong unknown genes, sequences flunking, and through PCR again. So we will be talking about polymerase chain reaction this time, what we call DNA sequencing and copying. So, and these genes isolated from reverse genetic approaches have limited their wider application. They have two main factors, no? They are functionally redundant. So ibig sabihin, madami super so pare-pareho lang ang kanilang mga functions, redundant na siya in the way, and simple gene sums in Arabidopsis have extensive duplication. And secondly, mutation in many genes may be highly pleiotropic, which is masking the role of a gene in a specific way. So reverse genetic is actually considered to be the major component ng functional toolbox, which is its role is assign biological function to gene through discovered large-scale sequencing programs. So transposon tagging also provides excellent alternative, for simple inheritance. So this is all about the functions of each genes to the phenotypic characteristics of crops, since we are dealing with plant genomics. Then we have also called transcriptional profiling. This analyze a few genes simultaneously, and these are one of the recent developments which allow parallel analysis of thousands of genes. So, so this involves gene expression patterns across a wide array ng cellular responses, phenotypes, and conditions, and expression profile, identifying genes, coordination and pathway, and their function. So this is again about functions of specific genes, identifying the the tags, the sequences. So this is all transcriptional profiling. So there are a lot of technologies, no? And if you read through this, so comparing also to animals, plants cannot move and have developed exquisite mechanisms to cope with changing environment conditions and biotic challenges that makes them very interesting to know it's what specific genes are responsible for that, for the transcription. So itong table na ito will show a comparison of microarray system. So we have um, copy DNA microarrays with direct or indirect spotting cDNA, and it's oligonucleotide microarray. So we will not deal so much on that. So as a whole, you just have to be to understand the gene function and the context of the organism biologically. And since what Arabidopsis is one of the first plants that have been mapped all throughout, so kompleto na no lahat ng characteristic ng Arabidopsis and its genes responsible for those are already mapped, no, na identify na. And there are also genomic tests and methodology available to the researchers. So, do you have any questions regarding plant genomics? So those are just
Wala, ma'am.
hopefully no kasi um even we're still student, I'm also in the academe, but even the scientists itself are still we are still in the understanding phase of this technology. So again, those were actually yung products na no DNA sequencing and copying. So let's understand how does DNA sequencing done, no? Wait lang. Hanap ko. Okay. So what is DNA sequence? Okay, so what is DNA sequence? Again, it is the process of determining the precise order ng nucleotides within a DNA molecule. So you remember in the previous discussion, we talked about DNA and RNA, the making of proteins, the transcription and translation. No? And it includes any method or technology. Yun na nga, we have a lot of yung kanina reverse genetics, we have EST, no? And so on. Those are some of the advancements or procedures in identifying, sequencing, and tagging DNAs or nucleotides. So, by the way, I have given you your your final exam na. So I hope you will enjoy guessing. So it's all about guessing. So if you have questions regarding kasi naka-blank lang siya, it's called analogy exam. And since you have one description and you should match the description with the given letter or blank. So I will give you examples later. So going back to DNA sequence, so any method that is used to determine the order of the four bases nucleotides, we have the AGCT, adenine, guanine, cytosine, and thymine, which is the strand of DNA. So if you remember before, we have um, the what do you call that? Yung squares of the different the DNA. Oh my God, I'm lost of words now. So I will just show you later ha, kaya nakalimot ko. Any of you remember DNA? Anyway. So DNA extraction is when you, of course, extract the DNA, no? And this is done through chemical treatments which cause the cells and nuclei to burst. So if you remember your DNA, yung spooling DNA, so extraction um, in your laboratory exercises, if you have done, so I remember some of you have done it, no? So don't worry if you don't really get the perfect white DNA, yung spooled DNA, at least you have just, try it, no? Na if makakuha mo, then good. If not, it's not really end of the world, no? So that is just a practical exercise you can do later, if you have all the facilities na. So the DNA is sticky and can be pulled out using yung stick or yung ating rod. So this is called DNA spooling. So ito na yung uh, picture ng spooled DNA ninyo. Wala lang siyang sip-on, no? So and inside of it are actually restriction cutting DNA code restriction enzymes, at specific sequences. So we have here a row to show you some of the restriction enzymes which cut DNA at specific sequences. And this is very useful to divide DNA into manageable fragments. So we also have another technology called electrophoresis. So you remember that term? It's where DNA can be separated based on the size and charge. So the phosphate groups are negatively charged. So yung mga phosphate groups natin, DNA is placed in a gel and electricity is run through like this. So in the electrophoresis, the negative DNA moves towards the positive end. So in this gel, this is the gel that is used in electrophoresis. So if negative siya, it will move towards positive end. The red electrode and the black electrode. And then if DNA is drop here, then it will move. Then the smaller fragments will move farther and faster. So you will see a sample na next in the end, a sample result. So in the restriction enzymes, this is the first step is this is your DNA sample, and in the first step is the restriction enzymes cleave the DNA into the smaller segments of various sizes, and then these are put in here, and the DNA segments are loaded into the wells in a porous gel. So itong gel are just made of yung kanang gel, no, na yung kinakain nga natin na gel, yung say tawagan na Micah? Micah?
Jelly Ace, ma'am.
Yes.
Yes, ma'am.
Gulaman, ma'am.
Yes, gulaman or agar. No? So then the gel floats in a buffer solution with a chamber between two electrodes. So ito yung negative and positive. So it will goes to the positive And then when an electric current is passed through the chamber, DNA fragments move toward the positively charged cathode na. And then the smaller DNA segments move faster and farther than the larger DNA. So it makes sense kasi pag payat ka, paspas ka mo dagan, di ba? Pag tambok ka or bugat ka, maglisod ka og dagan. So it means that kasi yung lahat ng chemicals natin are based on molecular sizes, no, or molecular weight. So you are identified as protein, you are identified as let's say glutathione or valine or whatever enzyme if you belong to this molecular weight. So ayan na sila, no. From here they will run through. So these are the smallest DNA and these are bigger DNA. So in electrophoresis as well, this separates DNA fragments based on size, so as I just have said. And the difference in size exists because each fragment, after being cut by the restriction enzymes, has different number of base pairs. So kaya pala yun sa kanilang phosphate groups. Then the DNA travels through the gel according to their sizes as shown earlier, and the gel is porous and acts like a sieve with smaller segments, being able to navigate around the pores easier than the larger segments. So this is actually the reason behind why smaller segments will travel farther and larger segments will not. No? So this also another illustration, no, of the electrophoresis. So always remember electrophoresis. So again, this is the gel, no, and this is the cathode or negative ion and the anode, the positive ion. It is where the mixture of DNA molecules of different sizes are dropped here or loaded here, and then the gel glass plates. So I remember on my when I went to UP, I have done this several times, no, and it's just really very easy, and I can show you, hopefully I can show you video if we still have time left. And so these are the result, no, the different DNA segments, smaller with smaller and larger sizes, so the longer molecule and the shorter molecule. So again, these are the different parts. Again, no, we call them well. It's where we upload the samples, and there are actually buffer here, yung mga chemicals that will help in running through the electrode. And these are again made of gel, yung agar or yung sabi nyo kanina jelly ace or gulaman. And so these are the smaller fragments that run through, and it is embedded in a plastic frame. So these large DNAs compared to small DNA, the small DNA will continue to move further and faster in the trade than the longer DNA. So it's the negative and positive pole. Besides this will represent the current which is passed through the gel to provide the separation. So this is actually what look like in real, in photo. So these are the large fragments and the small fragments after gel staining. So i-stain mo pa yan bago sila makaano. So now we proceed to DNA sequencing. So in a single DNA, we could make a lot of copies of that. No, we can multiply its copies. Then they are placed in a test tube and run through a DNA polymerase. So a mixture of nucleotides is added, some of which have di molecules attached and which contain ATCG bases, adenine, thymine, and guanine has different color dye for each. So these are the steps. Yung dinucleotides and some regular ones are added, then the di molecules are large and stop the chain from growing. So the result is DNA fragments of multiple sizes with colors that can be identified. So of course, itong magka-color lahat, yellow, red, or blue, are of the same yung fragments or yung guanine, that thymine, and so on. Then after the gel separates the resulting fragments by size, we read the sequence from the bottom to the top. So from the bottom, ito yung makikita mong sequence nila. Now there is actually a computer that will generate the different phosphate, no, or the different bases, yung A, G, C, C. So ito na yung mga codes or sequence nila. No? And so again, it is read from bottom to the top. Yan. Bottom to the top. So how do you do the copy of DNA? So we do this by polymerase change. No? So it's just like, which is in short called PCR, a method of making many copies of pieces of DNA. So as you can see on the figure, yan. Para siyang photocopier actually, even the machine itself. So nakakita na ba mo og ka ng PCR in real time?
Wala pa, ma'am.
So yeah. So maybe it's so far, because I previously I worked with USEP and we have a PCR lab, and of course UP, and of course etong mga natin, the DOST, they should have one. Although wala pa man ko ka-visit, so I am not really very 100% sure kung meron talaga sila, but I hope they are supposed to have one, and even UP Min, and who else? So maybe the Department of Agriculture in the sa kuan sa may Kalinan ba na siya? So there are steps in copying DNA. First, it's placed in a small test tube. Then the DNA polymerase are being heated to high temperature. So you will see the figures later. And then DNA is heated to separate two strands. The primer or short pieces of DNA complementary to the end of the molecules to be copied are also added. So yung primer yung tawag sa mga pieces ng DNA, short pieces, which are molecules na ikukopya natin. So this figure shows how to copy the DNA. Yan. So this is the primer or the original DNA to be replicated. And then once it heated, number one is the denaturation at 94 to 96 degree Celsius. Ayan. And second is, we call them annealing at negative 68 degree Celsius. And then at third is the elongation at 72 degrees. So hindi natin yan nakikita inside the machine. They are just being heated, and then it will work on their own. No? Hindi yan natin makikita again. So again, in the first step, the primer or the DNA to be copied are heated at 290 to 100 degrees Celsius to separate the strands. So this is the first separation. Ito yung primer. And then we have the new DNA and the old DNA. So the DNA is quickly cooled. So sa second step naman, i-cool yan to 30 to 65 degrees Celsius, so lower than 90, to allow short single-strand primers to unheal to their complementary sequences. And then Third step is the solution is heated again to 6 to 70, higher than the steps 2. So DNA polymerase synthesizes new DNA strands, creating new double-stranded DNA mo. So magdo-double yan. And then again, the entire process or entire cycle is repeated again. And each time the cycle is repeated, the amount of target DNA doubles. No? Ayan. So again, these are the steps in copying DNA. So, ayan, I have this as already have mentioned. So the tube is cooled and DNA polymerase adds new base set to separate the genes. And then this is again another photo to show you the large amounts of DNA can be made from a small starting sample, your primer. So it can be generate from one copy to thousands or millions of copy. So this is also very synonymous when you say clone, no? To clone. This it means a collection of molecules or cells which are all identical to an original molecule or cell. Or to clone a gene is to make many copies of it, replicating it in a cultured bacteria, no? And the clone gene can be a normal copy of a gene or wild type, and can also be altered version of a gene we call the mutant. And to work directly with specific, okay, sorry, to work directly with specific genes, scientists prepare gene-sized pieces of DNA in identical copies. It's called DNA cloning. So this is again the technique for reproducing DNA fragments. A vector is required to carry the DNA fragments, and uh, the uh, into the host cell. Then this allows copy of any specific part of DNA or RNA sequence to be selected among many others, which will produce unlimited amount. No? So this technique is the first stage ng genetic engineering when you alter the genes already. So this includes production ng DNA libraries, polymerase chain reaction, and DNA sequencing. And so for the basics, you just need the DNA to be cloned, anong vector, so to carry, maintain, and replicate clone gene in the host cell. So that could be most probably bacteria. Kasi mas madaling ano ang bacteria, mag replicate. So we also have restriction enzymes. Ito yung magka-cut ng DNA. So restriction enzyme are particularly related to the type of course, DNA. And we also have the DNA ligase para to join foreign and vector DNA, recombinant DNA, and whose cell in which the recombinant DNA can replicate. So which kind of cell? So, and for the first step, again, the DNA is cut from the source organism using restriction enzyme, and this will recognize specific target sequence and cut down DNA into pieces near on that site. So this is the figure to show you. So step 2, the piece of DNA is pasted into the vector and the ends of DNA are joined with the vector DNA by ligation. So this is how ligation or mura po siya ganang i-cut na nimo, no? The usual ligation term. The vector is introduced into the host cell, often by a bacterium or yeast, by a process called transformation. So it's called transformation, where the host cells copy the vector DNA, doing with their own DNA, creating multiple copies of the inserted DNA. So the bacteria and even the yeast, yung yeast na ginagamit talaga natin for baking, can actually has the ability to copy the DNA, no, of of your primer or selected DNA to copy. So this is how in the figure show the bacterial transformation from the bacteria, then you're going to grow that into plates in a host cell environment or media. And then you select transgenic cells resistant to antibiotics or with gene of interest. So we have two types, transformed and non-transformed cell, and then they are put in the nutrient milieu with ampicillin, the antibiotic, and then grown, and then you will have the ampicillin-resistant colonies. And fifth, culturing the selected clones. So you select the clones and then further culture them into separate fields, plates. This makes them to culture good nutritional medium without any limiting resources. No? So this you just transfer them just like when you separate bacteria or fungi no na ano sila culture and plate the bacteria on agar containing ampicillin and X-gal and incubate until they grow. So actually we have the advantages and disadvantages of cloning. Of course, for advantages, this allows scientists to study genetic disorder more closely and can grow replacement organs in many small animals. And larger and more resilient food can be grown, but it's very expensive, highly inefficient sometimes. Cloned animals tend to have compromised immune functions, higher rates of infection, and tend to be larger size, therefore causing them to have shortened lifespan. So usually if you are clone, mas ang immunity mo or yung function mo is lower, and then because science have not perfected it yet, aside from it's very inefficient and very expensive, it also have shorter lifespan. But right now actually we don't have an actually a especially kasi meron na tayong ethics, no? Protocol na hindi tayo pwede mag-clone ng mga animals and especially human beings. But then we also have project from other countries, especially China na may hilig na mag break ng protocol. I'm not saying no, sorry. But I have in the module 5 cloning ethics here. I can show you some na nakita ko just also from the websites na they have done so far in terms of cloning. So in the next topic we're going to discuss first how do they extract DNA in animals, because it's also different although the process is just the same, and we will do a lot of and you can see here the different cloning done mostly kasi pag for plants cloning is just parang common lang siya. So I actually have videos already. Okay. So I actually have videos already before on PCR. So this is 7 minutes. Okay. So let's watch this 3 minutes video before mag times up ta. Okay, so in just 4 hours, you have millions already of DNA copied through the DNA to the polymerase chain reaction. So do you have any questions regarding our topic for this session? Anyone? Do you have any question? So we're less than a minute, so anytime will be cut off. And thank you for listening and joining me with our class today. And hopefully we can finish all the topics that we have. No, and next time we're going to talk about animal DNA extraction in our module 3. So any questions so far? Mary Rose? Ikaw na mo Miss Intrams? Mary Rose? So wala na may pangutana?
Wala na,
Wala,
ma'am.
Sure mo. So what is at least your reaction no on this topic? So far have you undergone, so is it your first time to see what